Hi everybody and welcome to Marine Fishes Part 2. I'm not going to blah 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 with you with all the intro stuff that I usually do, so let's just get to it and let's just start talking about Fishes Part 2. Now we've learned about the taxonomy of fishes, we went into different groups of fishes, now we're going to be learning about general characteristics of fishes. Now some of these are going to be repetitive, so this one might go a little bit quicker because, again, I've already pretty much talked about all these major topics before, at least brought them up. So most of this we should be just a general review, so hopefully you guys are going through this and you're kind of remembering all the stuff that we've been talking about so far. So without further ado, let's go ahead and just jump right in to Marine Fishes Part 2. Alright, so let's talk about physical structure for a minute. Now, I kind of keep mentioning this whole like structure determines function kind of theme throughout the semester, and that's because it really is true. So again, we're going to be looking at some of these different fishes, and we're going to be able to kind of determine where they live and how they feed based on their body morphology. So the first one we're going to talk about are tuna. So remember we talked about the caudal peduncle, that's the area between the body and the actual tail or the caudal fin. Caudal peduncle. If it's really tiny and skinny, you're probably going to be a fast swimmer like the tuners and the billfish. If it's a little broader and a little wider, that probably means you're going to be a really maneuverable fish. And therefore you can just flick your tail once and you're going to get that big burst of quick movement, unlike a tuna, which again would give you that quick back and forth, back and forth, back and forth, back and forth to give you high sustained speed. So different behaviors are based on different, body morphologies, and really it's the other way around, but I want you guys to learn them kind of simultaneously. All right. So let's go ahead and just jump forward. Again, we've already talked about the caudal peduncle. We've already talked about tuna and billfish. This gives them a greater thrust when you have a really tiny caudal peduncle, and it gives you greater maneuverability when you have a larger caudal peduncle. So more things like angelfish, butterfly fish, they have a really wide, big, thick caudal peduncle because, again, they're not fast swimmers. They're not out there swimming in the oceans for extended periods of time. They're kind of cruising around the reef, but oh, here comes a predator. I need to dodge in and duck in behind a piece of coral. You need to do that quickly, and therefore that big, thick caudal peduncle gives you that nice maneuverability. So again, if you have a streamlined body, like think of a tuna, we're going to see one in just a second, but think of a tuna, nice streamlined body, almost looks like a torpedo, very thin caudal peduncle, very forked tail. These are all characteristics of a highly maneuverable, highly speed-oriented fish, okay? So these guys are doing long sustained swimming for long periods of time and have bursts of just extreme speeds like the tuna and stuff. Now, vice versa, if you don't have that torpedo-like, really stream-like body morphology, then you're probably not one of the fast swimmers, and that's okay, because you're not out in the middle of the ocean, you're probably hanging out by a reef, and that's, for you, that's going to be totally fine. So in that case, that body morphology works great for what you need it to be, which is just, highly maneuverable. And sometimes you don't want to be either, right? You're not highly maneuverable, but you're not fast. And that's because sometimes you just want to plain hide. So something like a stonefish or a toadfish or even like a flatfish, these guys aren't worried about their maneuverability. They're not worried about how fast they can go. They're literally just like, I'm going to stay here and hide in plain sight because you can't see me anyway. So it really doesn't matter how fast I am. So taking a look at a couple of these different body morphologies, again, we can see this torpedo streamline body morphology. We can see this very tiny caudal peduncle right here, and this nice crescent-shaped tail. That's how you can tell this guy is going to be a very fast, sustained swimmer. Now, some of the other ones are going to be, none of these are your highly maneuverable body morphologies, but you can see on all of them, again, thicker caudal peduncles, thicker caudal peduncles, and that's really just for like one or two quick bursts of the tail, and that's going to give you a good distance between you and your potential predator, and therefore, even though it's just a quick, still you're going to be able to get away from that predator fast enough to avoid predation, hopefully. Now, these are the kind of guys who don't really worry so much about maneuverability. Right, the trunkfish, the flounderfish, the seahorse fish, the sea moth are like, I'm not trying to maneuver anything. And they're usually going to be either a little more bony like this guy or, spiky like this guy. So again, he's not really worried about outmaneuvering a predator. He's just going to use his general defenses as a way to avoid predation. So it's not either or. You're not a fast or maneuverable. Sometimes you're kind of a little bit of both, and sometimes you're totally neither. Now, when we talk about swimming patterns, fish usually exhibit this S shape back and forth. And this is something that you've probably seen, right? This back and forth and back and forth. If you think of a shark, right? Back and forth, same things like a tuna. And that's basically because they have these bands of muscles known as myometers. And essentially, these myometers are kind of striated or almost like striped, and it's the way that they're formed to actually get the most efficient movements for them with as little energy as possible. So this kind of like zigzag, almost chevron pattern back and forth actually helps these myometers. And again, it kind of moves them in that generalized back and forth S position. And it really depends on your morphology again. So it depends on what kind of fins you have. If you got that nice caudal peduncle, right, that really tiny one, you're going to do back and forth, back and forth, back and forth, back and forth, back and forth. It's still going to give you that S motion. It's just going to be much faster. If you have a wider caudal peduncle, then you're going to be doing more of, again, the general back and forth until you need to do that quick, I just darted around a coral reef real quick. And then some of them, again, more eel-like ones, you can definitely see that S snake-like back and forth. And this is all, again, the position of the muscles and the myometers. So that's what we can see right here. So it's always going to be that S. This is a clear S. This is a very clear S right here. This guy, again, he's going to just beat his little caudal peduncle, his tail, his caudal fin right here. And then he's going to use his fins, his pectoral fins, to kind of maneuver. This is also another thing with tuna and some of these billfish is really faster swimming fish that they have. They don't really have the dexterous fins like you see like your goldfish. You can kind of like turn and maneuver and go back and forth like this. Right? It's because he can move his fins in all sorts of different ways, and they're soft rays, so they can really just fold and bend, and he can do whatever he needs to. A tuna, they actually have more rigid fins, and that's because if you're at high speed and you put a little ray out there, it's just going to get shredded, and then you don't have a pectoral fin. So you want more of a rigid, almost like reinforced right here. So as you stick that out, right, it's going to give you a hard left or a hard right, but you're not going to break it off or tear it apart. So those would be more of like, not spines, but harder structures to actually create that durability in your fins so that you don't, get it ripped off. These are the myometers that we were talking about. See the chevron pattern, the back and forth pattern. Again, you've got it going along right here, and this is going to be found in most of your fishes. Remember that we have that outer white meat and that inner red meat. Not all fishes have that red meat because not all fishes are going to be doing long sustained swimming and therefore need all that extra blood in your muscles. But things like tuna, that's why you can find red meat tuna and white meat tuna, is because the outer white muscles are going to be, again, just less oxygenated because they don't really need to, more for everyday swimming, and that red muscle is going to give you the real, like, strong, like, gung-ho motion that they would need. Remember that these guys have no swim bladder, so if you're actually looking for lift, what they do is they take their pectoral fins and they actually tilt them slightly upwards. And it's not something that they're doing. They're not like, let me move it. Because sharks actually really can't move their pectorals at all. Fishes can kind of out and in and out and in. And, ooh, look, some of them can go like this. And some of them even hold on to rocks like this. Some of the rockfish. But sharks can't do that. Sharks basically have a very stiff pectoral fin. That's another difference between sharks and fishes. And therefore, their fins are always out, right? Their pectorals are always out. You don't see jaws going, hi, right? They're just always out and they're stiff. But they're slightly tilted upwards, like the wings on a plane. And this is actually something that kind of we got from nature, is just looking around and going, well, if they're negatively buoyant normally, they should sink, but they don't because their fins are tilted slightly upwards, giving them lift. So if they're slightly buoyant and you're going slightly upwards, you're going to get that nice plateau, and then you're going to be able to just swim straight. Whereas a fish, if they need lift, all they need to do is fill up their little swim bladder right there, and then literally they have lift. We have lift off. And this is exactly what this slide is talking about, how sharks do not have a swim bladder, but they do have a very oily liver, whereas fishes actually have that swim bladder that they can actually inflate and deflate if they need to. So again, the sharks are going to use that stiffened, hardened, slightly turned upwards pectoral fins to give them the lift that they need, unlike the fishes. This is exactly what we talked about right here. Oh, okay, so again, this is where I was talking about the fact that bony fishes are going to have that more maneuverability in their pectoral fins, whereas sharks are not. They don't have any. So they can turn their bodies and kind of go this way and turn their bodies and kind of go that way, but they don't have nearly as much maneuverability as a fish would, a bony fish, and that's because of these pectoral fins. So this is if you're ever wondering like how a sea lion can swim a shark. It's not that he's faster than a shark necessarily, but that sea lion is going to be, woo, just dancing around you because they have flippers that can go out and in, and really maneuverable ones too. Where sharks are like, hang on, give me a second, I gotta slowly turn left for a minute, right? So they can bend, right? Their bodies are really bendable, so they can bend and then swim in that direction, but they're still not nearly as maneuverable as these, as mammals or bony fishes. Most mammals, like sea lions in this example. All right, so let's talk a little bit about their fins. Again, their fins are going to vary depending on what kind of species you're talking about and kind of what their environment is and what they're going to need those fins for. You're going to have the more rigid fins if you're going to, again, be those fast swimmers that you need to put out real quick and take back in. You're going to be more flexible and more dexterous, right, if you're going to be one of those highly maneuverable fish that you need to kind of move around real quick. If you've ever seen our California State marine fish, the Garibaldi, the big, bright, beautiful orange one, there you can see them always doing this with their fins, almost kind of moving it very soft, like almost like little tiny fingers. And those are the soft rays that we talked about in the dorsal fins, right? The spines and the rays. The rays are always going to be a little bit more dexterous, a little more soft, a little bit more maneuverable than the hard and sharp spines. And that's, again, just depending on the species specific and what they're using it for. Are you being maneuverable? Are you a fast swimmer? Are you doing neither? Maybe you're like the seahorse and you're actually just kind of hanging out and they have terrible pectoral fins. They're like this. Have you ever seen a seahorse in like a big current? They're just kind of like flopping all over the place, holding on with their little, hooked little tail. So again, it really depends on where they live and what they're doing with these fins. Now, fish gills. These are pretty cool. These are arranged in several different parts, and yes, you will need to know the parts of the fish gills. So you have the, I'm going to go into the pictures, the diagram of it in just a second, but I want you guys to know the different parts of it. So there's the arch, there's the rakers, there's the filaments, all sorts of parts that we need to know that are really, really important. And there's not even that many of them. There's like 4. All right, so the gills are actually really, really efficient in what they're doing. And you guys should probably know what they're doing, which is just taking oxygen out of the water. So that's what's the most important function for gills, right? We're removing the oxygen from the water. We're bathing our cells in it, which we desperately need for things like cellular respiration. And then we're removing that CO2 that we created via that cellular respiration and this metabolic waste and whatever else we might need to get rid of. So this is all happening via respiration, and their respiration is happening via these gills. So they do need oxygen, and they do need to expel out their CO2. All right, so here's exactly what they look like. So this right here would be the gill arch, okay, that little arch part right here, which is also right here. So this is the gill arch. So if we were to open up the operculum, imagine we just completely took the operculum off. This is what it would look like on the inside. You can see the gill arch right here that's curved right there. Okay, each one of these coming off of it, these are going to be the filaments, okay? Each one of these filaments has little lamellae on them. Okay, this is going to increase surface area. It's really important to increase surface area in your gills. That's going to increase the amount of diffusion that's possible. Okay, diffusion happens just simply from one side to the other. So imagine this is my arm, and I'm trying to diffuse something from in my arm to out, right? Well, if this is all that I had, then you could only basically do it on this top layer. But if you now increase the surface layer, right, increase the surface area, okay, you can think of these as kind of like the lamellae on each one of the filaments, so it already looks like this, and then you add little bumps on top of this. That's what's going on with the filaments versus the lamellae. It's just adding tons more surface area. So now I can actually do diffusion here, or here, or here, or here, or here, or here, or wherever I want to, because there's a lot more surface area. So surface area is very important to allow natural diffusion to occur. This means you're going to get more oxygen. This means you're going to remove more CO2. You're going to do all the things that you really need to do via these gills. So essentially what you have right here is each one of these lamellae have inside of them little capillaries. And capillaries is where the diffusion is happening. It's essentially very, very thin veins and arteries, so thin that they can actually have a single layer of oxygen that diffuses straight through. Okay, so really, really, really thin, and that allows for, in these guys, countercurrent circulation. So countercurrent circulation is a very important topic that we really haven't talked about yet. I've kind of breezed through some of the other ones in this lecture, but this one we have not talked about. And essentially what that means is your blood and the water are going two different directions. So you're getting this counter current circulation, meaning maybe you have a higher oxygen concentration here and a lower oxygen concentration here. That means that oxygen is going to be able to diffuse through. Great, that's what you need. Okay, but you're also going to get things like the CO2 being able to dissolve from here to here. So you're going to be able to get rid of that. But it actually happens kind of twice. So essentially you come in with a higher oxygen concentration, and then by the time you go, you're still doing diffusion. You're still being able to pull out whatever oxygen out of that water that you possibly can. So it's really, really efficient. In fact, I'm going to show you guys a link to a video on countercurrent circulation that I want you guys to watch, because I really can't even demonstrate with, I don't know, hands and fingers to really show you what's going on here, but it's absolutely amazing, and it's what makes fish so efficient at what they're doing. Efficient? Sorry, terrible dad joke in the middle of my lecture. But really we allow all of this diffusion to occur based on this countercurrent circulation. So it's really amazing. So you have one side which is being passed over the deoxygenated blood, which is going to be pulling oxygen. The other one's passing over the oxygenated blood, which is going to be removing CO2. But essentially, they're still just doing diffusion at all times in this side and on this side as they go back. So it's really amazing how efficient these fish are at doing this countercurrent circulation. I think we got everything here. So we have the gill arch. Again, that's going to be this part right here. Each one of these is a gill raker. So each one of these little spikes right here is known as the gill raker. Each one of these is known as a filament, right? This little U-shaped filament right here. And then each one of these bumps on top of it, which are going to be basically this guy right here, increases our surface area even further. Those are the lamellae. And then where diffusion actually is occurring is inside the capillaries. All right, moving on to mouth structures. So this is actually where, again, structure begets function, my favorite thing to say. So you can, again, just based on what they look like, you can kind of start to deduce on how they're going to be able to feed. So if we have a big mouth full of teeth, you're probably going to be a grasper, right? You're going to and capture something with your teeth. Think of like sharks and barracuda and stuff like that. If you have a long skinny beak, like maybe like a plucker beak, maybe you're going to be plucking individual polyps, right, off of that coral reef, like the stony corals, like the cnidarians. If you have a big crushing mouth with not maybe a lot of teeth, but kind of like plates for the crushing, maybe you're going to eat that coral whole. Maybe you're just going to take a huge bite out of the coral and then smash it up to get those little polyps and stuff like that, like some of the parrotfish. All sorts of different types of mouths to actually use in different ways. So even though there's all these different reef fishes living on the reef, a lot of them are using different resources or in slightly different ways. And remember that resource partitioning we talked about allows for maximum species diversity, and that's exactly what we want. So we see the barracuda with the bitey teeth, right? The capturing the teeth. We have the long, skinny little bill right there. That's going to be of a butterfly fish or an angelfish. We have the parrotfish right here. These are kind of the hard, crushy fish. Remember that parrotfish eat coral reefs, and most of sand is coral. So when you're walking barefoot, it's coming out of a parrotfish. We have the slip mouth, which is kind of sneaky. He basically has his mouth tucked in, but then he goes, right? It's a little ambush right there. We have the herring. We didn't talk about the herring, but the herring actually has, what you can't see is a super big mouth. But he actually kind of tucks it up and folds it up. So what happens is he has what's called a gape and suck mouth, where you actually kind of go, boo, and then you open up really big and you suck in all this water so that the prey that might have been this far away from you and your mouth is all the way over here, you can actually extend forward and hence gape and suck. So you open real wide and suck in all that water and your prey too. So that would be another feeding mechanism for these guys. Now, the position of the mouth is also very important. Is it here? Is it here? Is it here? Right? All of these different mouth positions can actually allude to where you live. Right? If my mouth is here, I probably live on the bottom. Right? So that my prey is going to be swimming above me. So as they swim above me, I can go with my mouth look like a flatfish or a flounder. If my mouth is down here, I probably feed along the bottom, meaning I'm going to be swimming along going, right? And if my mouth is all the way out here, which is known as terminal or at the end of the face, I'm probably going to be a chaser, right? I'm probably going to chase you down because I'm going to want to as much as I could. So again, even the position on your face is really determinant of your feeding behaviors. Now, digestion, we talked about increasing surface area, which is very, very important but in respiration, but it's also very important in digestion as well. So you always want to increase the amount of nutrients that you're getting out of your food, right? Food is scarce sometimes. Some of these organisms don't eat every single day. If you're starving and you eat something, you need to make sure you get all the calories out of it that you possibly can. And that's where your digestive system is actually going to come into play. So you need a digestive system that can actually handle it. And sometimes they have unique ways of doing so. Say also you're an ambush predator or just any predator and you're like, cool, I'm going to eat you, but you're almost the size of me. Well, I'm still going to try to eat you. In that case, you want probably an expandable stomach to be able to take in all the food that you just ate. Right? You don't know when you're going to eat again. Therefore, you need to make sure that you pack in all this food that you possibly can. And if that means eating someone who's almost your size, yeah, it's not really a problem if you have an expandable stomach. What else? They also have stretch receptors in their stomach, which is pretty cool. These basically, when stretched out, kind of trigger the stomach to be like, oh, there's food in here. Let's start working. Because otherwise, you don't want your stomach trying to churn and trying to break down food that's not there. So essentially, they're sitting dormant until your stomach's like, ding, oh, stretch receptors turned on. I have food now. Let's start breaking it down. And that's actually when they start to go in and start kneading it and stuff like that. Because really, that's what your stomach does. It kind of like rolls in on itself and kind of mechanically breaks up your food. Most people think it's chemically. Really, a lot of that has happening in the duodenum. Some of it's happening with the HCl, the hydrochloric acid, and stuff like that in the stomach. But really, that's what your stomach is doing. It's essentially rolling in on itself, trying to mash up that food. Which is why your tummy grumbles when you're hungry. Because it's essentially trying to mash up nothing. And it's like, why is there nothing in here? That's why that vibrates. You're like, yeah, fun fact. And these guys don't actually have it because they have stretch receptors. So they don't need their tummy to grumble because it doesn't need to do anything until the stretch receptors have actually triggered a chemical reaction to actually start to make that mechanical digestion occur. Let's see. The intestine, the pyloric cecum, the pancreas, the liver, all of these are helping to do chemical digestion, right? Adding these digestive enzymes to help break down that food. Remember, chopping it up into small bits that we possibly can so that we can get maximum nutrient absorption. Now, the intestines of carnivorous organisms are usually going to be a little bit straighter and a little bit shorter than herbivores. And that's because protein is a lot easier to break down than things like cellulose. So cellulose or other plant material that you might find in zooplankton or algae and stuff like that, it's actually pretty hard. In fact, it's still pretty hard for us to digest. We have what's called a cecum, and that actually is used to help break down cellulose and plant materials and stuff like that. But we don't need a ton of plant materials, so our cecums are pretty small. But if you think about a koala that all day, every day is just eating eucalyptus, and eucalyptus is a hardy plant, they have a super long circular cecum, and that's because it increases the surface area so that you can actually have maximum absorption, maximum breakdown of that cellulose, so you can get a maximum absorption of things like your nutrients and stuff that you would actually need, like ATP. So again, all of that is just to increase the surface area to make sure that we're getting as much out of our food as we possibly can. So carnivores, a little bit shorter, a little bit straighter. Herbivores, a little bit longer, and sometimes a little bit more of this going on to again break that down. Because if we just roll along here, but if we actually increase the surface area, then we can get maximum absorption and breakdown. Keep in mind, though, it does need to stay in your intestines a little bit longer if you are an herbivore, because again, your intestines are longer usually, and two, it takes a lot of time to break down that cellulose, and therefore, if you increase the surface area, you increase the amount of breakdown that you can do, and therefore increase the amount of nutrients that you can absorb. So taking a look inside, we can actually see a couple of differences between bony fishes and cartilaginous fishes, or sharks and fishes. So one of the most noticeable things that we can see is in the shark, we actually have what's called a spiral valve inside the intestines, or a spiral intestines. And really, if you look at it, like his stomach leading to the intestines, leading out to the cloaca, or, essentially his anus, is very short, and therefore he doesn't have a lot of surface area to do the absorption. And sharks notoriously don't eat very often, and therefore need to absorb all the nutrients that they possibly can. So inside of his short intestine, they actually have this spiral, almost like, you know, a garage. Think of like a parking garage where you have to go around and around and around and around and around to get out. It's the same kind of thing. You're maximizing the space in such a small area. So you're increasing the surface area in a very, very small distance. So this allows them to increase this absorption of these nutrients and stuff in compact areas. Now, similar over here, we have something that's known as the pyloric ceca. And really, this is, again, just like our cecum, this is very common for fishes who could be herbivores or even omnivores. If you're going to have any kind of cellulose, any kind of plant material inside of you, including algae, right, you're going to want that pyloric ceca to help you digest some of that, because that's going to be some nasty stuff, and it's going to be very hard for you to actually digest. Notice the size of the liver as well. Remember, we have a big oily liver here that helps with flotation, helps with buoyancy. Much smaller liver right here. Otherwise, the rest of them are going to be pretty similar when it comes to the structure of the digestive system, at least between the two. But spiral valve in the sharks and pyloric ceca in the bony fishes. Now, moving right along to the circulatory system, we kind of talked about it when we talked about the countercurrent circulation, right? Water's going one way, blood's going the other, so you maximize the diffusion all the way throughout the entire bend of the gill arch and the filament, which is really amazing. But they're not so great at the circulation because, well, they were around a lot longer than we were. And therefore, we've kind of developed a little bit more efficient circulatory system than they have. With our 4-chambered heart versus a fish's 2-chamber heart. But otherwise, they're pretty much the same. They have a closed circulatory system, so they have veins and arteries and blood circulating within. Let's see, they do have capillaries, and that's actually where the diffusion of the oxygen and the CO2 is actually going to occur. Remember, we saw that, those little tiny capillaries where you go from one big vein or an artery, and essentially you get really, really thin and start passing over, and that allows simple diffusion to occur just within the thin membrane of those capillaries. Let's see. We can actually diffuse in many different locations, not just in our gills, but also some of the tissues of the body. So again, we're having diffusion occur because we're sucking out oxygen, but we also need to get rid of that CO2. So the oxygen's got to go to all of our cells, and the CO2's got to come out of all of our cells. So again, this diffusion is not just happening in the gills, but it's actually happening in all of our tissues because that's where we need to remove the CO2 from. That's where we need to get the oxygen from. You know, even though your gills are doing the oxygen diffusion into your bloodstream still needs to take that oxygen all over your body. And if that's to your fin all the way down at the end of your body, that's where it needs to go. Just like our lungs are way up here, but I still need to get, you know, oxygen down to my little toe kind of thing. And that's what we can look at here, several different capillaries of the body. We have the head region right here, very important to keep the brain oxygenated. The gills, where all the good stuff is happening. The 2-chambered heart right here, which really is just returning deoxygenated blood to the gills. And then your vital organs all the way to the rest of your body and down through your tail, where you're going to have different capillaries to basically get that oxygen and that CO2 to different parts of your body where it might be needed. Now, the senses are very important for fishes. They aren't necessarily that smart. Therefore, they can't necessarily think ahead, but it's all really got to be instinctual because their predators aren't that smart either, but they're really fast. And they've evolved together over long periods of time. So the better the predator gets, the better the prey becomes at not being predated on, not being prey. There it is. But then the predator gets better at, you know, being able to catch that prey so he doesn't starve. So again, it's this evolutionary trend that the predators and the prey are constantly at it, and therefore senses are key. Eyesight, smell, hearing. Yes, things do have ears in the ocean. Weird. All of these things are really, really important to help you not be prey or to help you find and get prey, right? It's not just all about not being eaten. Sometimes you actually have to eat so you don't die. Very important. So we have the lateral line. Again, this is something we already talked about. We essentially have this jelly-filled little cup with cells inside called neuromasts. And these neuromasts essentially are just cells that are sending these signals to your brain, like neuromast, to help you instantaneously make decisions that you're not even conscious of. So if there's a fish on your right side and your lateral line gets detected, it's like, can you tell? Your lateral line detects it. It sends a signal through your neuromast cell, which is actually what picks up the signal. And it's going to send it immediately to your brain. Your brain's going to go left, someone's on our right, get out of here. And this is how fish can make instantaneous decisions, especially when they're schooling. They all do it at once. And this is all just because of their lateral line. So it helps them avoid predation. It helps them move in groups. It helps them detect when somebody else is near them, including their surroundings, all sorts of really good stuff. Remember, a school is really important, so safety in numbers when it comes to a big school of fish. Because, well, for one, a predator is going to have a really hard time picking one of you and focusing on you. So it's like, it's hard to focus this. There's so many. But two, also, like I said, safety in numbers. And three, you're going to kind of look like this big mass. So from afar, a predator might not know what you are. A predator's like, I don't know, that's kind of bigger than me. Kind of looks like this really big organism. I don't know. So it might be a deterrence just because your sheer bait ball size is big enough to make you look like your own big scary organism so that the predator might not want to come by. But here's how that works. So we have the lateral line up top. We have the ampullae of Lorenzini down below. Remember, sharks have the lateral line and the ampullae of Lorenzini, but fishes only have the lateral line. So this is our little neuromass cell right in here. See the little tiny neuromass cells? This is basically our little jelly cup. And that's going to vibrate just slightly, triggering the little neuromass cells to send that signal down the nerves to the actual brain. These are essentially little scales right here, and the lateral line is going and has little tiny openings all the way down the lateral line. And usually, depending on the fish species, it's kind of shaped differently. Sometimes it only goes halfway, sometimes it goes the whole way, sometimes it's surrounded by their face. It really depends on the species and whether you school or not. Schooling fishes are going to have a lot more surface area when it comes to lateral lines than a fish that doesn't, you know. Ampullae Lorenzini is very, very similar. Essentially, what you have right here is you have these tiny little pores, basically just adjacent to the scales, just like these guys right here. These tiny little openings. It's going to allow the water to travel in through this little jelly-filled canal that's going to kind of vibrate just a little bit, triggering the ampullae of Lorenzini, which is going to send its signal down through the nerve cell and out to your brain. So very, very similar structures, but, you know, kind of functionally just slightly different. And again, sharks do have both. You can see right here, these are the pink little dots are the ampullae of Lorenzini. The blue ones is your lateral line, whereas the fishes only have the lateral line, or I should say the bony fishes. Now, we already talked about this. This organ essentially is detecting electrical signals. So this is electrical impulses that your heart will give off, your muscles will give off. Every living organism gives off electrical impulses, and therefore this ampullae of Lorenzini can actually detect said impulses, detect those little vibrations, and then determine that you're actually there and you're actually alive. Now, schooling we already mentioned. Again, this is done by a ton of schools. This is about safety in numbers. This is about deterring predators. It's also like, hey, the chances of you getting eaten, that's the whole safety in numbers thing. As well as, it's really confusing for predators to actually pick one of you out. It's like, I don't know, I had that one, but now it's gone, and now I could have focused on this one, and now, and you're just trying to grab at something, and you usually you end up missing everything. So, safety in numbers really actually does work when it comes to schooling. We already talked about how you might appear much larger and therefore, again, deter those predators a little bit. We talked a lot about the schooling in the past, and therefore, you know, kind of blowing right through this. There we go. Territoriality. This is a cool one in fishes. So some fishes will actually make little nests, and some fishes will just have their little territories, and they're fiercely defendant of their territories. They're like, get out of here! It makes sense if you have a little nest. Essentially, you might have eggs or fertilized eggs or something that you're taking care of right there, and therefore you want to protect it. You don't want someone coming over and eating your eggs. Why are you protecting them if someone's going to eat them? You don't want other potential mates coming over and trying to fertilize your eggs. Those are your eggs. So a lot of this territoriality is done by males when they're protecting their nest. Some females do this as well, but common in males. The Garibaldi that we talked about earlier, the California state marine fish, Hypsipops rubicundus, super fun scientific name to say. These guys are notorious for being territorial. In fact, I had a girlfriend who did a study on this, and she actually tested, because they're in competition for mates, and so the male will kind of like garden his little area, and then the female will come lay her eggs. He will then fertilize the eggs, and she swims away as he protects those eggs. So he's very, very territorial over them. And I had a girlfriend who did a research project on them where she would show fishes like a block of equal size with the same bright orange color, and they'd like lose it, and they'd start like flaring out their fins, and they'd start mouth gesturing, and they'd get all like angry face. And then you show them like a big green block, which represents a different species, and they'd be like, huh. And then you show them a little orange block with blue spots on it, because juvenile Garibaldi have blue spots on it, to let the adults know, hey, don't beat me up, I'm just a juvenile, I'm not going to fertilize your eggs, don't hurt me. So she showed the little blue box with the blue dots on it, or the little orange blocks with the blue dots on them, and they're like, but then you showed a little orange block with no blue dots on it, and it doesn't matter the size, no blue dots, and these Garibaldi go crazy, and they're like, get out of here! And again, these blue dots is an evolutionary adaptation. Well, it's an advantage that they have, that the little babies don't get beat up because the adults are so territorial as long as they have the blue dots. Once they lose their little blue dots, then they're sexually mature, and then again, they get aggressive with each other. So if you've never seen a Garibaldi and the baby blue dots, oh, they're so bright and beautiful and colorful. It's crazy. All right, so again, some of this posturing is like really, like they open their mouth and kind of look like they're kissing each other. They flare their fins out. They get aggressive. They try to look as big as possible. Most of this is just kind of like a what? You want to go? They don't actually really want to go. They might get hurt. You might get hurt. They don't really want to fight. Now, will they? Of course. The territorial animals get territorial sometimes, and they will fight and beat each other up. We're going to see all about that when we get to mammals. Mammals can get pretty brutal, but they don't really want to. So most of the time, it's just posturing. It's just gesturing. But sometimes they can actually get pretty crazy. All right. Now, some of these behaviors include migratory behaviors. And that means from going from one place to the other. Now, we have some that will live in salt water during most of their life, and then they'll travel back to fresh water to have their babies. We will have some that will live in freshwater their whole life, but will travel out to sea to have their babies. And so it really depends on where you are first and where you reproduce into what category you fall under. So anadromous would be like salmon. Okay, they're born in freshwater, they live their life out to sea, and then they come back to the same stream that they were born in to have their babies. Okay, that is anadromous. Catadromous is the other way around. You live at sea your whole life, and then you come to the freshwater to reproduce, and maybe you go back. Right? Salmon don't go back. Some of these guys can go back and forth. Most of the time when you go, it's pretty much a one-way trip. You're there to reproduce and not much else afterwards. So, benefit, your offspring have protected areas where there's no competition. Negative, you die after you reproduce once. So you better make it count. So, like some of these eels that we actually talked about, some that actually go to the Sargasso Sea to have their babies. So they'll basically spend most of their time, living over here in Europe, and they'll actually travel to Sargasso Sea. Some of these guys will actually live in the freshwater areas and then also travel to Sargasso Sea, some of these American eels. But you don't always have to go from freshwater to saltwater. You can go from saltwater to saltwater. It really just depends on your species and where you're migrating to. And we're going to see a lot of that when it comes to mammals too, especially whales. And some of the larger sharks do it too, like great whites migrate big time. We don't exactly know where they reproduce, but we do know that they migrate every year if you're a male and every other year if you're a female. So what else are you doing? You got to be pupping, right? One of the things we're trying to figure out. Science! All right, reproduction in fishes. This is something that we have talked a lot about, so I'm definitely going to blow through this real quick. Some fish are males and females, some fish are males and females, and some fish go from male to female. Now, it's rare that you actually have simultaneous hermaphrodites, but you can actually have this. But really what you have in fishes, if you are going to be a hermaphrodite, is a sequential hermaphrodite. You go from one to the other. So again, just like the catadromous and anadromous, these are protandry and protogyny. Okay, so these are starting off as one sex and then moving to the other. So protandry or proandy is what I always remembered it as, is first male. So this always starts out first as a male and then transitions later on to a female. Now protogyny or protogyny, I don't know how you want to say that one. That means female first and then you're going to transition on to a male later. So when it comes to protogyny, we have the California sheephead off of our coast. And if you see the females, they're small and pink and kind of grayish, not really colorful. And if you see the males, they got this like big flat head. They got this huge like dumpy jaw. I call them hillbilly fish because they kind of look like they're underweight and like 3 teeth. But the males look vastly different from the females. But every once in a while, you'll see a female that kind of looks like she's kind of like almost a male, and that's a transitioning female. So the male will control the harem, right? And he basically will mate with all the females, and he'll keep all the other males away, right? And there usually aren't any males because all the males are still females. And then when he gets removed, either eaten or he dies or he gets fished off, the largest female will then go, well, there's no males around, but I still need to reproduce. And then she will transition into a male. And then she'll start reproducing with all the females. So either way, it's a win-win. It increases your reproductive odds because, well, you're one and then you're the other. So if you can't mate one way, then you can totally mate the other way and still works out. So usually these, again, are some kind of social cues, like the removal of the largest male will trigger the largest female then to transition over. We already talked about this. The harems. Now, depending on, like, we talked about this when it comes to sharks, but fish can do this as well. It's mostly when we're talking about fishes, we're talking about the bony fishes too. Some are viviparous, some are live-bearing. Some are oviparous, these are the egg layers, and some are ovoviviparous, meaning they lay eggs on the inside, but they hatch, and then, therefore, you give live birth. Right, remember that's kind of the combination of the two. All right, we also have broadcast spawning. It's actually probably the most common in the entire ocean way of reproducing is broadcast spawning. And that's no different among fishes. There's a lot of fishes that go out and broadcast spawn. You release your sperm, I release my eggs at the same time, boom, never see you again, bye. Right, it makes sense though, zero parental care, zero effort, you can produce millions of eggs without actually giving them any, like, time and effort, and hope for the best. So maybe 10 out of a million survive, but then boom, you just had 10 offspring, just like that. Now you can go off and have 10 more in a couple more months. So, broadcast spawning. Some of the parents will actually protect, like the Garibaldi we talked about, will actually protect. Some of the interesting ones are the mouth brooders. They'll actually protect the eggs, but they keep them in the mouth. So the females will lay the eggs, the male will pick them up and store them in the mouth as they fertilize and grow in his mouth. In fact, that was even in Nemo. Remember the fish opens his mouth and all his kids come out? Yeah, mouth brooders. So successful because you are protecting your babies. Although you can't eat and don't get hungry because then you just swallowed all your kids. But it works out well. Very few of these guys, and under very rare conditions, can actually do what's called parthenogenesis, where essentially it's asexual reproduction with yourself. So you basically make a little clone of yourself. And in fact, there was a very famous case, I think it was a kimono dragon, in a zoo. Hadn't seen a male for like 30 years. All of a sudden, boom, she gives birth. And again, just like Jurassic Park taught us, the original Jurassic Park, life finds a way. So if she's like, hey, my goal in life is to have a baby and there's no men around, I'm just going to have a baby. So genetically, it's just a copy of her, and that's known as parthenogenesis. So when conditions maybe aren't great, but you still need to reproduce, and you didn't have a male around, parthenogenesis. Sometimes, say if you're a broadcast spawner, and it's really important that you get these eggs out, what you gotta do is you gotta time it correctly. So you gotta time it at the right time. And so sometimes there's mating habits or mating behaviors involved. And so we're gonna talk about that right now. Sometimes these guys are actually gonna change color. They're gonna basically flash their colors at you and say, hey, I'm ready. Sometimes they're actually gonna come up to you and start poking you. And sometimes it's poking at your genital regions. And that's to let you know that they're ready. And so you really have to reproduce at the same time if you're gonna be a broadcast spawner, because otherwise, if you really release and then it just all floated away and I released over here, that's not going to work. Right? So you really got to do it at the same time. So mating behaviors are pretty common in certain species. And so what you can see right here is, say this is a female and she's just cruising along. Then this male comes up and he starts to do some kind of ridiculous display in front of you. Kind of sounds like real life, right? The male's trying to get your attention. So then he's going to follow you, and he's going to kind of follow you, and then, okay, maybe you're hitting it off, all right. And then he's going to start kind of nudging you, and really what he does is he nudges you kind of upwards in the water column. This is kind of to take you away and take you out of the area so that you're no longer, you're no longer fair game to any fish that goes by. Like, you're kind of taken. You also want to stay away from other predators, so you want to kind of get up here where there's nobody here. And then what's going to happen is essentially poking and prodding and twisting and rolling, and then at one point, you're just going to kind of sort of be like a mounting, and then you're going to have the release. Because again, you want the genital openings to be as close as you possibly can to do the release together to ensure that at least some of your millions of babies are actually going to get fertilized. So very important to actually have some of these mating behaviors so that we can actually have fertilization of your eggs. Otherwise, what's the whole point, right? Now, other organisms will just skip this whole broadcast spawning nonsense and just go for direct copulation. So this is a shark, and we already talked about this. Remember, this is a female right here, and this is a male. You can see his claspers hanging down. And he will use these claspers just kind of like an actual penis, and he will kind of insert his, and this is really just a fin, it's a modified fin, but he'll insert his fin or his clasper inside her genital opening and release his sperm inside so that we really definitely get fertilization from this type of copulation. This is just broadcast spawners. If you don't want to do it like one-on-one, even if you're broadcast spawning, sometimes they all just get into a frenzy and they're like, and they all just kind of release at once. So you'll see a lot of fishes and they get kind of crazy and they start moving around and darting around and darting around. And then it's like one triggers and then all of a sudden you're swimming in like a cloud and you're like, that is sperm and eggs, go the other way. But it worked great for them. Notice this is also a little bit darker here. Usually this is happening around dusk or just afterwards, and that's because you just let out a lot of food, and there's going to be a ton of other organisms waiting for you to do so, and probably waiting to eat your eggs. And therefore you don't want that to happen, and therefore you need to, right? You don't want them to just start feasting on it. You want to do it when it's dark, and maybe all the other organisms are kind of gone for the day, and you're like, shh, let's go, go. Right? So not everybody gets eaten. Now, this chart is a really great chart. It's kind of a summary of all the things we've been talking about, from the hagfishes to the lampreys, from the rays, the skates, the sharks, the ratfishes. We didn't talk about the coelacanths. The coelacanths are really cool. They're one of the oldest fish and thought to be extinct like 1,000 years ago, but totally still around. And then finally your bony fishes like your tuna. So this is an amazing chart to kind of give you guys an overview of all the things that we've been talking about. So highly recommend that you guys study off of this one right here. Is that it? Are we done? Is this another lecture in the books? Yes, it is, and don't feel guilty. Right? Because we are moving right along to the rest of the chordates. We've got a bunch of fun stuff coming up, so make sure that you guys are studying for your practicals. Make sure that you guys are completing your labs and your homeworks and whatever else that we have. And I hope you guys have a wonderful rest of your night, rest of your day, whatever you're doing, or whatever time it is, or what day it is. And I will see you guys all very soon. And take care of yourselves, and have a good one.